Hi, I'm Becky Crean, Executive Director of Clinical Development at Ionis Pharmaceuticals. And first, I'd just like to say thank you to the Angelman Syndrome Foundation for inviting us to provide an update to the community virtually. We've been involved with the community for many years now, and it really is remarkable to see how so many advances have been made year after year towards developing a treatment for individuals living with Angelman syndrome. And as we've recently seen, there's been some setbacks along the way and disappointments, and that's not uncommon in drug development. It really is part of the process. It's hard to go through, but it is to be expected at times. But Ionis, together with our partners at Biogen, remain committed to developing our ASO, which is Ion 582, and we continue to make great progress each year in our program. And all of our scientists and researchers back at Ionis and Biogen are really 100% dedicated towards these efforts. So I'm excited to be here to tell you where we're at today. So many of you may be familiar with Ionis, but I realize there may be some new people listening in. So I'll spend just a few minutes introducing you to Ionis and then speak specifically to our Angelman program. So just by way of background, we were founded in 1989, really with a single mission. We wanted to turn antisense oligonucleotides, or ASOs as we call them, into a therapy. And since the discovery of ASOs, for the last 30 years, about 100% of our focus has been on developing and advancing this technology. We're committed to this technology in a way that really might seem stubborn to some. Our sole purpose is to discover and study medicines to find more effective therapies for people with rare and common disease. And while most of our focus continues to be on developing ASOs, we've now broadened our scope to include additional types of RNA targeting therapies in addition to ASOs. So our technology is now working. We've had some good success along the way. We've brought 4 transformational medicines to the market and to the patients who need them. We have a pipeline full of ASOs in development, many in the clinic, some in the research, for the treatment of a variety of neurological diseases like Parkinson's, Alzheimer's, ALS, Angelman syndrome. In terms of the company itself, we've about doubled in size since I began working here. We now have over 800 employees and still counting. We're headquartered in Southern California in a town called Carlsbad, which is where I'm located. But we also have offices in Boston and Dublin. So we're a global company now. I mentioned our robust pipeline and also our 4 marketed products. We developed Spinraza to treat spinal muscular atrophy. We've developed Tegsedi to treat polyneuropathy, a drug called Waylivra to treat FMS. And finally, the most recently approved drug called Qalsody is an ASO designed to treat a very rare genetic form of ALS. So we have a long history and a lot of experience in developing genetic medicines, specifically to treat rare diseases caused by genetic abnormalities. So many of you may be aware of Spinraza, the life-changing medicine it's been for kids and families with SMA. So I want to spend just a few minutes talking about Spinraza because there are a lot of lessons we learned from our experience with developing Spinraza that we can and have been applying to developing our treatment for individuals with Angelman syndrome. So first and foremost is you really need to know the disease. SMA, like Angelman syndrome, is a rare monogenetic disease, meaning it's caused by a mutation in one single gene, but the symptoms are not all the same in each person. No 2 kids are alike, just like in Angelman. And so we need to really understand and know the disease very well so that we can develop a drug to treat it. That's why natural history studies are so important. They really do allow us to collect invaluable data to better understand the disease. Also, we have to know the patients with the disease and how they're medically managed, what types of medicines get prescribed, what types of therapies do they have. Standard of care and medical practice can be different depending on what parts of the world and even in the country you live in. We also need to make sure we talk with you, the families, so that we can understand what's important to you for a treatment. We can't just assume it. We need to ask the families, listen, and then incorporate what we can from that into the drugs we develop. Also, choosing the right endpoints is critical, really to be able to answer the question that your study's trying to answer, which typically is, does your drug do what you want or expect it to? Also, we can't take a one-size-fits-all approach. Rather, we need to find the right endpoints for the right patients. That's partly what we do in these early studies. We give a lot of tests so we can determine which ones work best for the population. Endpoints, as you know, can be pretty tricky. Measuring something that's easy to measure may not be meaningful, and what may be meaningful may be difficult to measure if the endpoints aren't sensitive enough to detect changes. And also, which sites are involved in your study is critical. So not every clinician can be a PI in a clinical trial. They may be great clinicians, but it's a little bit different to be a PI in a drug trial. Sites need to have the infrastructure set up to handle complex studies and complex dosing regimens, like with intrathecal dosing for ASOs. They need to have the study staff skilled at administering these endpoints we just talked about, and they need to be skilled at it as well. So we work very closely with sites. It really is an investment and a partnership together, and it can take some time to build that relationship so that you can have really important, open and frequent communications and address problems that come up, which we know they do. So there's a lot involved in choosing PIs and study sites, and it's important. It's an important decision when you're running a drug program. So Ionis has been working on a treatment for Angelman syndrome for about the last 9 or 10 years. And it really started with the very early work with Dr. Art Baudet at Baylor College and his team. They tested one of our ASOs in the Angelman mouse model and really made the first discovery that it improved the symptoms caused by the loss of the UBE3A protein. The father's UBE3A copy, as you know, is normally silenced in neurons. And so our ASO unsilences dad's copy of the gene in order to produce the UBE3A protein in the brain. So we've really taken our 30 plus years of experience to discover Ion 582, and we've focused on developing the safest and most effective ASO that we could. And safety really is our top priority. So our HALOS clinical trial is the first trial in humans, and its primary focus is on testing the safety and tolerability of the drug. So I'd like to explain just a little bit about what an ASO is. Many of you are aware, but to do that, I have to take you first back to your sort of high school biology days so you can understand the central dogma of life, which in its simplest form is DNA makes RNA, which makes protein. That's how the machine works. It does it over and over again. And sometimes when things go wrong on the gene, then the copy or the RNA carries forward that wrong message. And so our system then makes too much of a protein or too little of a protein, and that causes the disease or the disease symptoms. So our ASOs are small strings of nucleotides, and they bind to the RNA, which in theory corrects the mistake that it has on it. In turn, it'll either produce more of the protein our bodies need or less of it, depending on what the issue is. So ASOs are considered disease modification drugs because it targets the source of the disease, unlike, say, a small molecule drug, which works on the protein and targets more of the symptoms of the disease. So we need to get these ASOs into the brain, but they're too big to cross the blood-brain barrier easily. So to get them there, we deliver them by injection into the cerebral spinal fluid, CSF, by a lumbar puncture into the intrathecal space, which is near the lower back. So we have a lot of experience doing this with other ASOs. We've treated over 9,000 patients with this method. So it's a very safe and well-tolerated method. So as I mentioned, Ionis really pioneered the development of antisense oligonucleotides, and it's something we live and breathe every day. Lots of other companies have tried it and said it would never work over the last 30 years, but we stuck with it. And as I mentioned, we've brought some really important ASO medicines to patients who need them. Now other companies have dipped their toe into the ASO water, so to speak, and are also developing them. But not all ASOs are the same. So in our HALOS trial, we're evaluating an ASO, as I mentioned, called ION582, that's specifically designed to stop the silencing mechanism on the paternal UBE3A gene so that in turn it can upregulate the UBE3A protein in the brain. It's called it's a type of ASO that's called a 2' Mo ASO, which is a specific type of chemical modification. So this type of gapmer, as it's called, it has the modifications at the two ends, and then the DNA is in the middle. And what this does is it activates something called an RNase H cleaving mechanism to target the antisense, which silences the gene. So not all chemical modifications are the same. As I mentioned, there's MOs like ION582, there's LNAs, which is the type of chemical modification used in the Ultragenic and Roche compound. There's O-methyls, there's many others, but each type of modification comes with different levels of potency, different levels of tolerability. Some chemical modifications have a better safety profile than others, so they all have their own unique characteristics based on the type of chemical modification they have. So MOs are designed for greater potency for reducing the target RNA and for their increased duration of action, which means how long they last in the body and also how well they're tolerated. And it's really because of these specific properties that Ionis develops MOs for drugs designed to go into the brain. So how did we find ION582? Well, it was identified after extensive screening, first in cells and animals. We started by screening over 3,000 ASOs that bind to a particular region on the antisense transcript. And from there, we picked the best ones to test in transgenic mice, which have the human gene inserted into them. We tested them in Angelman mouse models and also iPSC neurons. And from there, we took the best ones that came out of all those testings and then tested a group in large animals, non-human primates. And from there, we selected ION582 out of over 3,000 ASOs because it was the safest and most effective molecule for taking into humans. It has a very good potency. It's been well tolerated in multiple animal studies. And actually, in those animal studies, we tested at much higher doses and greater frequency than we use in the clinic. So they have very good safety profiles when they come out of this extensive screening process. So that's a little bit about what ION582 is, how we discovered it. And now I'll tell you a little bit about how we're using it in the HALOS trial. So this is a really important trial. It's the first phase of the clinical trial process for developing this ASO. The trial is designed to answer a very specific question about ION582, and it's the first trial in humans. And because of that, it's a small trial looking at the safety of different dose levels given over a relatively short period of time. And we're testing it in individuals living with Angelman syndrome. So we initiated this trial about a year ago. The trial's currently ongoing. The ASO is administered, as I mentioned, with an intrathecal injection into the spinal canal, which is the space around the spine. It is with a lumbar puncture, and when kids get this injection, they'll be given sedation to make them sleep during the short time that it takes to give the drug. So this trial is a combination of a phase 1 and a phase 2, and each individual will receive several doses of the ASO over a period of about 3 months, and everyone receives drug. There's no placebo in this first trial. So HALOS enrolls a broad age range, including young kids up to adults, but different age groups come in and are staggered and enter the study at different times. During the trial, children and families are given tests to measure the things that we know are most important to you, like talking, walking, sleep, learning information. Some of these tests are done in the clinic, some are done at home. Our primary goal in this first study is to assess safety of the drug at different dose levels and to make sure that it can be tolerated. This study will also give us the information we need to select the best dose and dosing regimen that we can test in future trials. So a really important trial that we're running. As I mentioned, HALOS is a small trial. But we have a very broad reach across the globe. We have sites in 6 countries. All of them are open and active. Having more sites in such a small trial does mean that each site will only enroll a small number of families. But we also think it's important to test the drug in as many geographical areas as we can, because we need to understand if the drug behaves differently in different segments of the population. So how's it going? Well, the steps in drug development can seem really slow and long. It's true. It's a long and involved process. It's not unusual for this process to take up to 20 years for a drug to go from the very early discovery process through all phases of clinical development and to be approved for use in individuals. But each year we make progress. We learn more information and we get closer to a treatment. We've been running Halos, as I mentioned, for a little over a year now, and we're actually right where we should be. And really thanks to all of you, the families, the sites, and the clinicians for your interest and willingness to be in the trial. We understand that it's a significant commitment with no guarantee that your child will receive any benefit from being in the trial, but really because of you, we continue to advance and make progress, and the study is progressing as planned. So very, very happy to report that. So for the Halos trial, it's designed so that a small group of individuals with Angelman syndrome are enrolled at the same dose level. And then after several doses of the drug are given in that cohort, then we follow them for some time to gather the safety and clinical information. And once that's done and the data is collected, that we review that and it tells us whether it's safe to go to the next dose. And then we enroll a small group again, and that group gets dosed for a while. And then we look at the safety data. So it's a process, and this process keeps happening until all the dose levels are tested and all the different age groups are evaluated. So in the last year, we've been able to test multiple doses of ION582. And while we don't release study data while the trial is ongoing, we can say that all the doses that have been tested in the studies so far have been well tolerated. There's been no safety concerns or drug-related adverse events that anyone has experienced. Another thing we've made progress on in the past year is that we've been able to add the long-term extension to the trial. This means that once patients finish the first few doses in the beginning, then they continue receiving ION582 for an additional 12 months. So that's called the long-term extension. So now the Halos trial has 2 parts. Part 1's called the MAD portion, multiple ascending dose portion, which, as I mentioned, just means different dose levels are tested in different groups. That data tells us which doses and dosing intervals are best for long-term dosing. Now that the long-term extension has been added, it's called Part 2. Primary goal is still to test the safety and tolerability of the compound, but over a much longer period of time. This part will also help us determine the best dose and dose interval that provide the best clinical benefit depending on the different ages, if it's deletion or mutation genotypes. So this part will include the additional 12 months of treatment, and we'll look at different dosing intervals over that time. So we've made great progress over the past year. And as I mentioned at the beginning, Ionis and Biogen truly remain committed to developing a treatment for individuals living with Angelman syndrome. It may seem like it's taken a long time, but we need to make sure we're thoughtful and thorough at each stage of this process. We want to make sure that we first and foremost determine if the drug is safe before moving to the next phase. So to answer that question, we need to first complete the Halos trial. We need to complete both parts 1 and 2. So the study will continue to run through the rest of 2023. We do plan to include the entire Angelman community in this process. So we do plan to extend the age range and test different mutations. We'll do that in the future. This will be driven by data from this Halos trial. I don't have more information on that now, other than to say that once we have the data to inform us, we'll include them. So in the next year, we also plan to continue analyzing the UBE3A and the CSF samples collected in the natural history study. An important part of this is not only understanding levels of UBE3A protein in the CSF, but also how those levels may change over time. So this will help us understand any changes we may see in a treatment trial, and if they're due to the treatment or just natural variation over time. So all of this information and all of this work planned is aimed at determining the safest and most effective doses of Ion 582 that can be used to be tested in the next phase of development for the compound. And as I mentioned before, this is a very sort of systematic approach. Each phase and each bit of data we collect is important. It's necessary in developing a treatment for Angelman syndrome. So I'm really happy to report the progress we've made and how well things are going. But really with that, I just want to express my sincere appreciation for your time today. All of us at Ionis and Biogen are deeply committed to the work we do on behalf of the Angelman community, and we've made great progress, and we couldn't have done it without you. So we thank you for your commitment to doing this together. So thank you.